Okay, so in this lecture, we're going to be looking at the organisms that are studied in microbiology. So let's get started. So microbes, as we have mentioned already in the previous lecture, are very, very small. We measure them in units of micrometers and nanometers. A micrometer is 1/1,000,000th of a meter. A nanometer is 1/1,000,000th, that's billion with a B, 1/1,000,000th of a meter. So these are super, super small organisms. For instance, and the book makes the point that something on the order of 500 bacteria could fit onto one of the periods that is, in the punctuation of your textbook. So super small things, some smaller than others, viruses certainly being at the very, very smallest end of things, and the parasitic animals that we're going to discuss are at the larger end of things. So let's start by looking at the viruses. Viruses are considered to be non-living particles because they're not built of cells. So one tenet of biology, it's called the cell theory, is that all living organisms are built of cells, and because they don't have all of the characteristics that we associate with a cell, then viruses are generally, by most folks, considered to be non-living. And they are the smallest of microbes. We can only see them when we look at them using electron microscopes, the most powerful of the microscopic tools that we have at our command. And what a virus is a length of nucleic acid, and that might be DNA, what we think about as the genetic material of the cell, of a cell or of an organism. Or it could be a close cousin of DNA called RNA. DNA stands for deoxyribonucleic acid. RNA is ribonucleic acid, and it's another kind of nucleic acid, and we'll learn more about these later. And this nucleic acid is packaged up in a protein coat. So if we look at this bacteriophage, it's a viral type that infects bacterial cells, the DNA is held up in here in this area. You can't really see the red, so I'll put an arrow toward it. Here's where the DNA is held. And all this stuff, the shell that is around it, and here we see these pieces here, they are all proteins. And so, or they're built of protein. And so, what a virus is then is a piece of nucleic acid that's packaged up in a protein coat. And the way that viruses work is, as a virus attacks a cell, it injects or somehow gets its nucleic acid into the cell, and then the nucleic acid begins making proteins that will take over the cell reproductive capacity, and so the cell will then start producing new virus particles. And there's kind of two ways that this can kind of go down. One is that the cell just fills up with these virus particles, dies, and then bursts open, releasing new virus particles out. Or it could be that the cell continues to live, and it makes a low level of, just begins shedding off a few of the virus particles sort of constantly. And so two ways that can go down, and we are going to discuss this a lot more when we get to chapter 10. Okay, so those are the viruses. And then the next on our list to look at are the bacteria. Now, that term bacteria, with a little b, it refers to two very, very large groups of organisms. They are up at the domain level. So if you remember from high school biology, or maybe a college biology class that you've taken, we can classify living organisms based on that kingdom, phylum, class, order, family, genus, species thing. And so kingdoms used to be sort of the highest grouping, the most inclusive grouping of classification for organisms. And in the past 30 years or so, a new level of classification has been introduced that groups together kingdoms into large clusters or groups. And so there are two domains, and that's what we call that largest, the most inclusive level of a classification system is the domain. There are two domains that we refer to as bacteria with a little b bacteria. That is the domain bacteria. There's the big B bacteria. And the domain archaea. And that's how you say that. Anytime you see an AE, that AE there together next to each other, you say E. And so that is pronounced the domain archaea. And cells or bacteria from these two groups, the bacteria and the archaea, are both single-celled, and they lack nuclei and internal structures that are built from membrane material, sort of like the cell membrane, but inside the cell. So they don't have those things. And if you recall from the previous lecture, we termed these groups prokaryotes. Again, I'm just introducing that term to you. We're gonna hit a lot more, with a lot more vigor in some coming chapters. So the groups are different, though, which is why they're clustered into two different domains, and the differences are very significant. So even though they're both prokaryotic groups, the molecules that are found in the membranes and in the cell walls of the archaea and the bacteria are very different. And so the structure of those structures, the construction, let's say, of those structures of the membranes and the cell walls is real different, built of different building blocks. And when you look at the structures that they use for motility, they're called flagella. They're like these long, slender extensions off of the cell that they use to either pull themselves or propel themselves. They're really different. So they have different ways of moving around, and the environments that they can inhabit are very different. So all of this, plus a lot more, a lot more that we're not even going to be able to get to in this class. A lot of their physiology is very different as well. So lots of structural differences, lots of physiological differences between organisms that are clustered under domain bacteria and those that are clustered under domain archaea. But when you look at them under the microscopes, there's a lot of similarities. And so because of that, we just have a tendency to call all of these single-celled, relatively simple cells bacteria with a little b. Okay, hopefully that makes sense. We can talk about it more in class if it's confusing. The algae are a third group. Algae are single and sometimes multicellular protists. Protists used to be a kingdom. So again, if you're old like me, you would remember a kingdom protista. We no longer think of it as a kingdom. It's actually a collection of kingdoms, as we've been able to do more and more, looking at the actual DNA that's found in these organisms. And so there's a whole bunch of kingdoms within this clustering that we call protists. And the algae are the ones that are photosynthetic. They are single-celled, and they have a nucleus, and they have internal structures built from plasma membrane. And so because of that, we group them into a domain called the eukarya, and we talk about them as eukaryotes. So that's the other term. And again, I'm introducing the term, but we're going to really dig deep into it in later chapters. They're found in salt and freshwater habitats or in moist soils. So they have to have water, though, to survive. And it turns out that there's only one genus within the algae grouping that we think of as, or that could be considered pathogenic. That is, disease-causing. And we're talking pathogenic in humans. And that genus is called Prototheca. So here is a light micrograph of Prototheca wickerhamii that can cause infections in humans, they are actually non-photosynthetic algae. So crazy to think about an alga that doesn't photosynthesize, but sure enough, there are some, and Prototheca is one of them. And there are some free-living algae that can affect human biology. They produce toxins that are dangerous to all vertebrates. And these are the algae that cause what are known as red tide or red tide events. And so when there's a big bloom of these algae, usually in salt water is where you find the organisms that can produce red tide, where they get into really high concentrations. That's what we describe as a bloom of these red tide events. When they're in high concentrations, they produce together collectively enough of the toxins that people who are on the beach or standing nearby can breathe in enough of them that they can be hurt. And any vertebrate organisms that are swimming and using the water can be killed by it. So red tide events frequently cause large fish kills, but they can affect humans as well. Okay, so the algae, single-celled and sometimes multicellular protists, but the ones that are concerning to humans are all single-celled. The fungi, so this is a kingdom. They are also eukaryotic. The fungi include yeasts, molds, and what we think of as mushrooms. Mushrooms are the spore-producing, very obvious representatives of a much larger and much, but still frequently microscopic structures that are growing through the soils. So yeast, molds, mushrooms, fungi can be single-celled or they can be multicellular. The one that we're looking at here is definitely multicellular. And rather than getting energy from sunlight, the way that the algae do, they get their energy by absorbing nutrients across their cell walls from the environment around them. And because of this, they can take up habitation on the human body and can be pathogenic. So these four diseases here, ringworm, yeast infections, athlete's foot, jockitch, these are all fungal infections of the human skin or of the mucous membranes of the body, where the fungal cells are growing. Excuse me. Sorry about that. Excuse me. So they are all pathogens where they are living on the surface of the human body, and they're absorbing nutrients across their cell walls. And they have great, they play great importance to humans because some of them produce antibiotic chemicals that will attack and kill, or that are part of how we attack and kill bacterial infections. And so penicillin, for instance, is an example of this antibiotic chemical that we use as a means of fighting infection. And protozoa, then, are the fourth group. These are also single-celled organisms, and they are protists, so they belong in that sort of large clustering of kingdoms that we refer to as the protists. And they're non-photosynthetic. Here we see an amoeba that is taken, that photo is taken under a light micrograph. They are eukaryotic as well, meaning that they possess nuclei and membranous intracellular structures. And they tend to prey on smaller microorganisms that are in the watery environments and moist soils that they live in, or they're capable of absorbing nutrients from the fluids in their environment. We find them in, as I said, aquatic environments and moist soils, but some of them can act as internal parasites. So if you think about amoebic dysentery, for instance, or malaria, the sleeping sicknesses that we find in tropical countries, and even hiker's diarrhea, the Giardiasis. That's a hard one to say. I think I must have spelled that. Giardiasis. Yes, I spelled it right. It's caused by a species called Giardia. And we generally think about it as coming from drinking waters that have been contaminated with fecal material from other mammals, humans or grazing animals. But it can also come from eating raw vegetables, say from a salad bar, where the foods were prepared by someone who had not used proper sanitation. So those are all protozoan diseases or protozoan-caused diseases. And then the last group that we consider to be part of what we study as microbiologists are the parasitic animals that are found inside human and the bodies of other animals too. So these are macroscopic organisms, so they're not microscopic. We can see them with the naked eye. And there are two groups that we kind of cluster into this kind of odd grouping because of their ability to cause disease, to be pathogenic. The first are the helminths, and helminth is just an old-timey word for worm. And there are two groups that tend to be most common as parasites in humans, the roundworms and the flatworms. And they are internal parasites of the gut or of body organs. So flatworms tend to be like on the liver or the lung. We describe them, or there's some that are called liver flukes, others that are called lung flukes. And roundworm parasites tend to be in the digestive system, or they may insist in the muscle. Arthropods, these are the groups of animals that have a hard but jointed shell around their body, include species like lice and fleas, ticks, mosquitoes, and flies. So insects and arachnids are in that arthropod group, and they act as external parasites of the human body, and they are important not just because they are parasitic, but because they also act as vectors for many microbial diseases. So viral and bacterial diseases, as well as protozoan-caused diseases, like malaria, for instance, can all be transmitted by these kinds of arthropod organisms. So there you have it, the viruses, the bacteria, the algae, the protozoans, the fungi, and the macroscopic animal groups. So, as always, if you have questions, you know how to reach me, either in class or you can send me an email, and we can talk more about whatever questions you might have about these organisms. Okay, study well. Oh, I should just say, I forgot that there's one more piece here. So microbiologists, these are scientists who study microorganisms. Their research interests tend to be varied. I forgot that this lecture included something about microbiologists here. Their research interests are varied, and they are involved in such pursuits as protecting public health. So working as epidemiologists or as folks who are developing or studying about the life cycle of different microbial organisms that threaten humans. They might be involved in developing and testing antibiotics and other means of controlling microbial infection. They are definitely, we saw in the last chapter that there are a lot of chemicals that are involved in, or that are produced by microbes. And so developing the industrial uses of microbes is another kind of work that microbiologists might do. And then environmental microbiology is an important field where we look and where we study the microbes in their natural habitats and environments and how they interact with each other, looking at the life history of specific organisms. And then finally, there are microbiologists that are using microbes as tools for studying physiology, so for understanding how different cellular structures function. Okay, so there. Now I think we are done with this lecture, and you know what to do.